disease. Can I have the next slide, please? Can I have the next slide, please?
Yeah.
Thanks. So why, or what was actually, also, there's a second reason to actually also change from a lipid formulation to a so-called lipid-free technology, meaning it's not only targeting the surface, or actually surface receptor and then entering into the cell specifically and not passively. It is one of the major issues with lipid nanoparticle technology is the toxicity of these lipid formulations. That actually prevented a lot of drugs entering into the market, or even to in late stage clinical development, is because it was not possible to increase dose to get to a therapeutic dose in using the lipid nanoparticle technology. Other issues are it's complex to manufacturing. Secondly, stability issues, because these lipid formulations are very unstable and have to be kept at very low temperature. However, there are lots of now research to actually prevent the or to actually increase the stability of these lipid formulations. With lipid-free technologies, it is indeed these molecules are more or less have almost no toxicity, or at least very low toxicity, and can be applied to very high doses. And that actually led to all these kind of drugs, which I already indicated in the list in the slide before. And basically by that, we actually now can reach, with these lipid-free technologies, reach therapeutic windows to actually get from a RNA therapeutic to a real drug to be applied in treatment. Can I have the next slide, please? So how does that look like? So how does a typical oligo drug look like? And that's actually depicted on this slide. So we have from the left to the right side, we have on the left side have a ligand. In this instance, it's a sugar moiety, which more or less interacts with a cell surface receptor. The ligand has a distance to the oligo. That's via the so-called linker, which is depicted here is depicted here as a linker technology. You can use different type of linkers. They can be normal type linkers. They can be like it is shown here, like packed linkers. So basically, it is dependent on the cell which you want to address with your drug, which type of linker is more suitable. Then you can link it to the oligo. The oligo itself is basically more or less the drug. The rest of this, so linker and ligands, are basically the so-called formulation to bring the drug into the cell. And as it is shown here, you can basically, and that's what we actually offer and also been using, and Nassif already introduced that. That is now frequently used in producing antibody drug conjugates, meaning it is already a concept which is used in many therapeutics in the protein world, which we now actually also introduce into the oligo therapeutic world. Can I have the next slide? So how does that look like? So basically, when you come to BaseClick or to any other company who is actually producing these kind of drugs, so first, what we produce is the ligand. And here, the ligand is, in this instance, is a trimannose. And basically is we more or less conjugate or chemically the ligand to the linker. So at the end of the day, you have a ligand-linker moiety. That's the first step in your development. So basically, you first choose your ligand, meaning in this instance, it's a trimannose that will actually addressing specifically the MRC1 receptor on various type of cell types, in particular immune cells. And basically you conjugate that in this instance to a PEG linker, which actually carries a clickable group, the A site group. Then in the next step, you produce your oligo. And the oligo is more or less, is in general, you can actually use either antisense oligos as RNA oligos. You can use DNA. You can use DNA, RNA mixtures, whatever you like GATMA technology. Basically, the oligo, that's really up to you what kind of target you want to address. Then what we actually have, we have different type of partners. One is a drug partner, and the other one is the formulation partner, which not only formulates it and protects it, but it also actually as a ligand to actually address the receptor on the targeted cell which you want to target. And then you do the click reaction, and that's more or less because the click reaction is an orthogonal a reaction type, which more or less gives you one type of getting together, and in particular because it's very mild, and that's a concept of Barry Sharp. It's very mild. You can do it in aqueous, but also in lipid in the lipophilic environment. You can do it at low temperature. So basically, it's a very mild condition to attach your ligand group to your drug group. Can I have the next slide? So coming to what we offer at BaseClick, because as I'm not only a researcher, but I'm also the CEO of BaseClick, and what we actually have developed is a number of different ligands, which you can more or less buy directly from our web shop. We have synthesized various ligands like the azido PEG4 trimannose or the trigalnac, as well as an azide or as a DB2 group. However, you can come to us, and that's what Nasifa, my colleague Nasifa already indicated to you, is you can come to BaseClick and we can synthesize you whatever kind of ligand you want to have from us. I will show you a summary later. Can I have the next slide? Beside this ligand, we also offer you various types of linker molecules. Because I already indicated that not only the ligand composition is very important to address the receptor on the surface, but also the linker molecule is important, because linker actually bring the drug, so itself like a protein, or in our instance, the RNA, actually separates it distantly from the ligand for the targeting. And also when it then enters into the cell, it has to be still functional. So basically the linker, or choosing the linker, is a very important part of your drug development. And as it was shown in the introduction, we actually have a lot of experience in this. I will come now. Can I have the next slide?
Yeah, sure.
Oh, thanks. I will now come to some examples. Here was a collaboration with the company BioNTech in Germany, where we more or less looked at, is it possible to bring an RNA molecule specifically into dendritic cells? This is particularly important for cancer treatment, because the dendritic cells are the major presenting cells in cancer treatments. So basically what we published that in the Journal of Carbohydrate Chemistry recently. So basically what is shown here is we produced a molecule having, in this instance, the trimannose ligand, which is addressing the mannose receptor on the surface of macrophages or other kind of immune cells. And basically what we actually found out indeed is that when we used PBMC cells and basically then the sorry, no, actually, can you okay, thanks. So basically what is then actually is that we have shown that only in immature dendritic cells you get a good uptake. And that's basically shown in this in this slide, is more or less you see a dose-dependent trimannose uptake into premature dendritic cell. They are not taken up in PBMC cells appropriate, and also not in mature dendritic cells. So basically we have, in this paper, we actually described that in more detail, that indeed it is very important what type of ligand you take to actually address a certain type of cell, but also you can actually investigate and also get your targeting correct, because for cancer treatment you need immature dendritic cells which actually then mature for presentation. Can I have the next slide, please? I will actually, because there was one question, I will address that maybe a little bit later, because it was how does the release in the cell is materializing. I will actually come to that question probably in a minute. So basically is the second example is shown here. Here we again used an oligo, in this instance an RNA oligo, which we not only linked to the targeting sequence, it is also in this instant it's a trimannose to address the mannose receptor. We normally then, in this instance, we also put a dye on the other side. So basically you can couple twice on the 5' and 3' end to monitor. Basically the question here was, is it possible to when you conjugate the same oligo to different type of targeting sequences, so the ligands, is it possible to address in the same tissue different type of cells? And indeed, that is shown in this slide, is we coupled the RNA to a trimannose ligand, and we coupled it to a trigalnac ligand, so addressing two different type of receptors. One is a mannose receptor, the other one is the azole receptor. And what is shown in this slide is we injected that into mice and looked in particular in liver and in some other tissues as well. So what we basically see is, and that's shown when you have an unmodified RNA, that is not entering into, or we can hardly detect the oligo in any of the tissues, because as I already mentioned, unmodified and non-lipid formulated, because this is just unmodified naked RNA, is not entering into the cell. However, when you link the oligo to an trigalnac, then basically what you see is it enters specifically into the hepatocytes. And that's shown in B. There you basically more or less see that it is more, indeed just entering into the hepatocytes. When you actually use the trimannose, what you can see, and that's shown in C, is it enters exclusively into the copper cells in the liver. And basically, these are the so-called copper cells, are the macrophages, named the macrophages in the liver. When you actually use, and that's unmodified RNA with encapsulated into lipid formulation, you basically then, and that's shown in A, it's shown in A and D, is you get it everywhere. You get it into copper cells as well as in macrophages. That we published recently in Nature Communication. Can I have the next slide? Oh, okay. Now I'm sorry. I forgot to answer your question. Is how the release or the question was what was addressed is the release of the oligo, conjugated oligo in the cell. So basically, the oligos, when it binds to the ligand, it starts to have an endocytosis uptake of this ligand receptor conjugate. And then you have an endosome. And the endosome changes, when it actually enters into the cell, changes its features. It normally is, you get acidity, so it starts to actually lower the pH, and then the lowering of the pH leads to the release of the drug, because it disintegrates and then the drug is released. And that, as I mentioned, is a very important, that's an important step to get the drug into the cytoplasm or to its part in the cell where it should actually confer its therapeutic application. That is why we have to really modify heavily the RNA, because the acidity of or the endosome, when it becomes acidic, would destroy the oligo if it is not modified. Now I will actually have another molecule which I want to introduce, is indeed what was recognized to be usable for oligomeric. That's across the membrane of the endosome. Oh, sorry, there's another question. So I answered that question as well, although the Q&A is normally a little bit later. So basically is in the release of the out of the endosome is, as I mentioned, is what actually is in the typical pathway of the endosomatic uptake is as follows. It is actually, first, the uptake is in a lipid, double membrane lipid endosome. And as I said, then you have in the endosome, you have basically H+ pumps, and it what actually becomes acidic. So it goes down, even down as low as pH 2 in some of the endosomes. It's a special. There are some going to 4, 5, even down to 2, and then the lipid membrane starts to disintegrate via this, because it's a typical pathway to actually get certain cargoes into the cell, like natural molecules, not only drug molecules. So basic natural molecules. In particular, when you use the mannose receptor, that is actually, it takes up pathogens. Basically, and then acidity starts to, and then the pathogen is also starting to be disintegrating. However, the endosome is then more or less disintegrating, and by that releasing passively the oligo into, or also the mRNA, which we actually will show you here, also releases that into the cytoplasm. And from there, it can be then either have its function in the cytoplasm, or it can be even further transported to the nuclear, or as we also have a program, it even can be transported to to mitochondria.
Sorry for interrupting you, Thomas. I'd like to request all her attendances. So I see all the questions, and you'd like to answer the questions in our end session. So it would be nice if we can wait and answer the questions at the end. Would it be fine for you?
For me, it's fine. So I would then refrain.
Let's go ahead with the webinar.
Okay, now come with the mRNA. So mRNA is differently produced. It's enzymatically produced, or at least currently it's still enzymatically produced, although there are attempts also to do it chemically. However, these days they're all enzymatically produced, and it's more or less it's what we call a so-called in vitro transcription. That is normally you do it from a plasmid or from a PCR DNA template, and since we actually are not producer of these plasmids or of these PCR products or the templates, we actually have aligned with Thermo Fisher and Eurofins in this instance. So the template is produced, and then we do the in vitro transcription to produce the mRNA. And what we have developed, and that's depicted here, we have developed also modification technology using the click chemistry to modify also these mRNA and also to confer to mRNA similar features which we actually confer to oligos. And basically that's what we offer. We can actually produce your mRNA, we can modify it, and then we deliver it to you. Can I have the next slide? So basically, that's how an mRNA normally looks like. You have a cap structure, you have your UTRs, you have your untranslated part of the 3' 5' untranslated part, and you have your gene of interest, the ORF, and also what you actually have to get a decent type of translation into your protein product, you have also poly-A tail. And what we have developed, and that's more or less is the invention. We also have patents on that, and also published that in the paper in Biochem in 2020. Is we can modify the 5' end internally as well as the 3' end, and you can either purchase all these products from us, or we can actually introduce that for you into your mRNA and send you the mRNA already modified. Can I have the next slide? I just want to show you one example where we used an mRNA expressing, in this instance, YFP. And what we were looking for, and I already indicated that, we actually published part of it together with BioNTech, is what we more or less is we modified the mRNA with a trigalnac and trimannose at the 3' end of the mRNA. So basically is the same concept as I have shown you for the oligos. The same concept was we introduced also in mRNA. So basically is you have your mRNA produced, you attach to the 3' end, so the end of the poly-A tail, you attach, in this instance, a sugar moiety, and we attached also the trimannose and trigalnac, meaning hopefully we can also address different type of cells, but also wanted to check whether that makes it possible to, when you inject this kind of molecule, inject it into a mouse via IM, so intramuscular, would that be, because it's not encapsulated, would that mRNA, which is modified, survive and basically enters into certain tissues in the mouse without being destroyed? And indeed, what we can show here is we injected IM, so intramuscular, into the mouse, and what we extracted is a lymph node, one close to the lymph nodes, one close to the injection site, and on the other side of the leg. It was actually in the leg. And basically what you can see here is indeed the lymph nodes adjacent to the injection site there's a dose-dependent uptake of the molecule, which is depicted here. Whereas on the other side, you see some kind of uptake, but it's not as distinct as you can see with the close to the injection site, which means indeed the mRNA, although it's not encapsulated lipid formally, is able to travel into, in this instance, into immune competent tissue like the lymph node. Can I have the next slide? And that it brings me to the summary slide. Indeed, as I have shown you, we can actually couple any type of nucleic acid drug to a ligand, which then can target the appropriate receptor on the surface. And we have shown that. I showed it only for sugars in this talk. However, we have shown that for fatty acid, anandamide, siRNA, where we showed indeed that will enter not only into PBMC cells, but also we have shown that that is entering into neural tissue via the cannabidiol receptor. I've shown you the triGalNAc. We have done extensively research on glucose, so via the glucose receptor. We have used monomannose as well as trimannose. We have also shown for folate via the folate receptor entry into different cell types, and also have shown that dipeptides also actually entering into, for example, cancer PSMA positive cells. Thank you. Next slide. Thank you for your attention, because I already summarized that with the other slide, more or less, and I'm open for questions.
So I'll start with the questions here.
Yeah, I would ask the participants also to post your questions in the group so that we can answer them one by one.
Yeah,